Hello everyone. In the previous part we have discussed about type 1 receptor signaling. Now we discuss about type 2 receptor signaling. As we have read before that type 1 signaling is the receptors that present in cytosol, and type 2 is the receptors that present in nucleus. So here type 2 signaling we see here thyroxine, retinoic acid, these are the examples of type 2 receptors, and they are present in nucleus and they are bind with hormone response element site. So let us take an example as thyroxine. Here triiodothyronine is a signaling molecule. So triiodothyronine is a signaling molecule that enters into the plasma membrane, crossing the plasma membrane into the cytosol. So that means it is a nonpolar molecule. We have discussed before that signaling molecule are two types, nonpolar and polar. So here triiodothyronine is the non-polar signaling molecule, and it can cross the plasma membrane and enters into the cytosol, and it has also enters into the nucleus. Here you can see that the receptors are bind with co-repressor HDAC, that is histone diacetylase. So histone diacetylase is a co-repressor that repress the expression of genes. That is, there you can see that no expression of genes occurs, and the DNA is in heterochromatin condition. As I have told in the previous part that the DNA must be euchromatin for the response or for the expression, but there is DNA is in heterochromatin condition, that is more condensed. So we have to, for the expression we have to activate the receptor, and the HDAC co-repressor should be dissociated. So how can it happens? We have to activate HAT co-activator. So HAT co-activator recruitment can help in activation of these receptors. Here you can see that HAT co-activator recruited. After recruitment of HAT co-activator, that is histone acetyl transferase, those triiodothyronine signaling molecules are bind in the receptors, and the receptors are bind with the DNA in the hormone response element site. How it bind with DNA? As I have told before that they have zinc finger motifs, and those zinc finger motifs is a protein domain that controls gene expression binding with DNA, and they have hormone response element site is also I have told before that it is a DNA sequence that binds to the hormone receptors to regulate gene expression, and it allows the promoter to change their transcriptional rate in response to hormones. So there is some zinc finger motifs are attached to the hormone response element site in DNA, and signal is present there, and with the presence of signal and in the presence of euchromatin DNA, target gene expression can occur. This is the activation of nuclear receptors. So first we see that after entering of triiodothyronine into the cytosol, and then it enters into the nucleus. Here, due to binding of co-repressor HDAC histone diacetylase with the receptors, the thyroid hormone receptors are inactivated, and there is no expression of genes due to heterochromatin DNA. But when the recruitment of HAT occurs, then it is a co-activator that activates the receptor, and there is hormone response element site where the receptor binds with DNA, and this HAT, that is histone acetyl transferase, activates the receptor, and it further occurs target gene expression, and the DNA present is euchromatin, thus signal is present here and the target gene is expressed.